This study is titled REM Sleep Selectively Prunes and Maintains New Synapses in Development and Learning. This research study was completed by Wei Li, Lei Ma, Guang Yang, and Wen Biao Zhan. This journal article can be found in Nature Neuroscience and was published on January 16, 2017. The main topics that this study examines are those surrounding the effects of REM sleep. This includes synaptic pruning, neuroplasticity, motor cortex development, motor learning, and neuronal circuit development. The hypothesis made prior to experimentation is that REM, or rapid eye movement sleep, is vital to neuroplasticity and selective pruning, which allows for greater efficiency of neural processing. 3 aims are focused on in this study. The first is to discover whether or not REM sleep has an effect on neuroplasticity and synaptic pruning of dendritic spines in layer 5 pyramidal neurons in the mouse motor cortex. The second is to determine whether REM sleep plays a role in strengthening the remaining spines. The third aim that we will be observing is how calcium spikes in REM sleep increase the size and strengthens new spines. REM sleep is something that occurs during separate intervals of total sleep time and involves increased respiration, heightened brain activity, rapid or increased eye movement, and muscle relaxation. It has been proven that REM sleep accounts for up to 50% of total sleep time during early development, and its duration increases after learning later in life. Another important piece of information for understanding this study is that there is a strong direct correlation between size of dendritic spikes and synapse strength. It has also been proven that acquiring and learning new motor skills increases new spine formation in young mice. To the lower right of this slide, you can see an image showing the rotor rod test for mice, which is often used in the study for motor learning. There are 3 different experimental groups used for the experiments of the first 2 aims, and for the third experiment, the grouping is slightly different, but this will be explained in the results of this study. For the 3 experimental groups, these include ND, or non-deprived sleep mice, NREMD, which are non-REMD sleep disturbance mice, and REMD, which are REMD sleep deprived. There were also 2 age groups of mice tested towards the beginning of the study to show which group should best be used for each set of experiments. The first group are the 3-week-old male and female mice, also known as the P21 mice, and these were used for the developmental studies. The next group are the P30 mice, or the 4 to 5-week-old mice, and these were used for the motor learning experiments. The first experiment, which targeted the first aim, involved disturbing the mice's sleep through gentle handling, which woke the mice when they were entering REM sleep. It was known when the mice were entering REM sleep because electroencephalography and electromyography recordings signaled this. The next experiment involved using the same technique as the first experiment, but instead doing this from time 0 to 8 hours over 4 consecutive days in P21 or 3-week-old mice. The last experiment entailed using a 2-photon calcium imaging model to observe the spikes of apical dendrites in layer 5. These neurons expressed a calcium indicator called CGAMP6, which allow for calcium tracking. For the experiment, the mice's heads were restrained and they were also trained to run on a treadmill under the 2-photon microscope. For the first experiment, again, the 3 experimental groups are non-deprived mice, non-REM sleep-deprived mice, and the REM sleep-deprived mice. For the first figure, figure F, you can see that pruning is significantly higher in the non-deprived and non-REM sleep-deprived mice. This shows that the mice that did not receive REM cycled sleep were not able to synaptically prune at a rate such as that of the non-deprived and the non-REM deprived mice. Figure K and Figure M showed similar results in that synaptic pruning occurred at a lower rate in REM sleep-deprived mice compared to that of other groups. The distinction between these figures and Figure F is that there was a motor learning involved in the tests. Figure K shows the results after forward running, and Figure M shows what occurred after backwards running. Overall, the data shows that REM sleep plays an important role in pruning newly formed dendritic spines in the motor cortex. These next results show the changes in spine strength following motor learning patterns that were administered through the rotarod device. Both figures include the percent size change induced by each motor learning pattern, which was forward running and backward running. For Figure G, the size of non-deprived mice spines increased more than the REMD-deprived mice, especially during the second time interval over the 8 to 24 hours. Figure I shows the results after the backwards running motor learning test. Similarly, this shows that the size of the REMD dendritic spines experienced almost no size change compared to the other two control groups. In the end, this shows that REM sleep contributes to increased spine size and strength, since both are directly correlated. For the last set of results, we have different experimental groups. These include quiet awake, running, non-REM, REM, and REM plus MK801. In this experiment, MK801 was injected, and this is an NMDAR antagonist. This MK801 is used to tag synaptic channel proteins which allow the movement of calcium. Therefore, this can track the voltage changes in these channels. In both figures C and D, it is shown that during REM sleep, the larger fold changes in calcium spikes and the higher calcium peaks occur. This evidence from the third experiment clearly shows that peak size and fold changes in calcium voltage are greater in the mouse motor cortex during REM sleep. After reviewing all the data, it is clear that there is a relationship between REM sleep and the spine elimination ability, dendritic spine strength, and synapse strength of newly formed spines. This discovery is very important since neuroplasticity and development have a lot of influence in early formation and function of the brain. Something critical to take away from this experiment is that someone who is deprived of REM sleep could experience a deficit or lack of motor cortex learning. Since this process of motor learning is dependent on the brain's ability to selectively prune dendritic spines and keep what is important so the brain can focus when completing that task. REM sleep directly enables and benefits this pruning process. Each experiment observed both the effects in newly formed spines caused by motor learning and pre-existing spines. But for all the compiled results, it was evident that there was no effect whatsoever of REM sleep after motor training on the pre-existing spines. This involves effects on synaptic strength, synaptic pruning, and dendritic spine strength. In summary, rapid eye movement sleep is critical for optimum efficiency of neuroplasticity and pruning during development. By removing excessive synaptic plasticity, REM sleep may free up spaces in neural circuits to facilitate memory storage. This means that a lack of REM sleep could be detrimental to the process of selectivity that is required for motor learning. In the future, further experiments can be completed to observe what other effects REM sleep has on the brain. These could be to observe more long-term effects on deprived REM sleep and the chronic issues that could result from this, or to determine the effects of increased REM sleep on neurological plasticity and development.